Hey guys, it's Mrs. Olenichek, and today we're going to talk about the cell cycle and mitosis. We're going to focus on mitosis and really think about the process that cells go through to divide. Generally, the big question here we're looking at is how you get from this, a fertilized egg cell. So we all started off when one sperm fertilized an egg, and then somehow we get to this or something like this. And so we're made up of millions of cells, and all of those cells have all the exact same DNA in them. They're specialized to perform different functions, but the process of going from this, that fertilized egg, to this, a multicellular organism, is going to be really important. You might ask why we aren't just one big cell. Why do you have to have lots of different cells? And the idea is that large cells have issues with transport. As cells get bigger and bigger, it becomes more difficult to transport materials throughout the cell via diffusion. And so if you have lots of different little cells, you have a lot more surface area than if you were just one big, huge cell. And so this allows for more efficient diffusion of nutrients like oxygen and glucose. It also allows cells to get rid of waste products more efficiently. So even though it might seem like it'd be simpler to just be one big cell, we find that cells, when they get too big, they basically start poisoning themselves because it takes too long for materials to diffuse from the center out. All right. So cell division is just the process by which you have one cell dividing into two identical daughter cells. The key here is that you're getting two cells that are exactly the same as the parent cell, and that's going to be through mitosis. The whole cycle is kind of broken up into two chunks. You have interphase, and this is where the cell really gets ready to divide, and then you have mitosis, where you have division of the nucleus, and basically separating out the DNA into two new cells. Before we go any further, we should probably make sure to define some terms. When we talk about chromosomes, this is basically just your DNA. So we've all kind of seen these kind of images. This is what a condensed chromosome would look like. Normally your DNA is kind of opened up and it's not all organized and condensed into these kind of chromosomes so that you can use it. But when a cell gets ready to divide, it's going to condense it into this organized chromosome. Chromosomes are made up mainly of DNA. They're also wrapped around these things called histones. Each chromosome is going to have a matching pair. So you got one version from mom and one version from dad. You have homologous chromosomes. During interphase, those chromosomes are going to copy themselves, and then during mitosis, they're going to be separated out into two cells. Your DNA has pretty much all the instructions for building proteins and doing all the things that cells need to do, and so it's really important that you don't lose part of those instructions in the process. Another kind of set of terms that come up when we talk about chromosomes is the term haploid versus diploid. So if you look here, this represents a haploid chromosome set, and this represents the diploid. And so here you've got 1, 2, 3 chromosomes, and so we would say that the haploid number was 3. So this sperm and this egg would both be haploid. So this one would have, we'll say, 3 chromosomes, or if this is human, it's 23 chromosomes. And the sperm has the other half of that set, so it would have another 23 chromosomes. So when they come together, you form a diploid cell. So that's where you get the complete set of chromosomes, your diploid. So I remember haploid, H for half. You get 1/2 of the complete set. And sometimes you'll see that represented as an N. And then diploid is where you have the complete total set with both pairs. and that's usually represented by 2N. So we're really going to be focusing on diploid cells in this process. Haploid cells are really just your sex cells, so they would be the sperm and egg cells, and that's a different process to make those. That's going to be meiosis, but today we're going to just focus on mitosis. So once you've got that full diploid cell, how does it make copies of itself, and how does it make exact copies of itself? If we look at the kind of life cycle of any cell, we break it down into what we call the cell cycle. Now, interphase is going to be the longest phase. That's going to be all of this right here. Let me see if I could highlight that for you. So this whole thing is going to be interphase. And then over here, this is just the mitosis part. All right, let's see if I can get rid of that now. All right. So interphase is broken down into 3 little sets. So you have G1 is the first growth phase. I think of it as like the growth phase, but it's really gap 1. And this is where cells grow and do all of their cell things. Once they get a signal that it's time to divide for whatever reason, then they're going to start looking at the cell contents and getting ready to divide. Once they've done that, they're going to move into the synthesis phase, and that's where DNA makes a copy of itself. And then you have G2, which is the second growth or gap phase. And here's like kind of the double check, right? You're going to make sure that there's no errors in your chromosome copies. You're going to make sure that you've divided all of your organelles and you have everything you need. And there's going to be checkpoints throughout this process to make sure that the cell is ready to divide and that it doesn't have any mutations. We'll look more closely at interphase and the checkpoints there in another lecture. But if the cell's okay, if it passes all of its checkpoints, then you can move into mitosis. So mitosis is where you're going to divide the nucleus. So you've already copied the DNA in the S phase. Mitosis is just organizing and dividing and making sure that each of your new cells comes out with a complete set of DNA. We break that down into phases. I just remember PMAT, prophase, metaphase, anaphase, telophase. So it's like IPMAT would be interphase, prophase, metaphase, anaphase, telophase, and then you'd go back into interphase. But really mitosis just includes that prophase, metaphase, anaphase, and telophase. So prophase is going to be your first step. Here your chromosomes are visible. They start to condense. Something called the centrioles. So remember these from when we talked about plant and animal cells. Only animal cells are going to have visible centrioles. So they'll start migrating to the poles of the cell, kind of like the North Pole and South Pole. They start migrating to different ends of the cell. The nuclear membrane is going to disappear. The nucleolus disappears, and you have a spindle forming. So spindle are these fibers that are coming off over here. So these start to grow out of the centriole. So here's your centrioles, and you have spindle fibers starting to grow out of there. And these are kind of the things that you're going to see during prophase. Next, you have metaphase. This is where your chromosomes line up in the middle. So remember M for middle. And here we call it the metaphase plate or the equator. So kind of like we were saying, the poles, North Pole, South Pole. Now in metaphase, you have your chromosomes lining up single file by the equator. Next, you have anaphase. And this, I remember A for away. So here the chromatids are going to separate and move to the opposite poles. So you had your duplicated chromosome, and now one copy is going to move in this direction and one copy is going to move in the other direction. Then you get telophase. So now that you've got your chromosomes all the way at the other side, your nuclear envelope is going to start to reform. The nucleoli reappears and your spindle fibers disappear. You'll also see the centrioles duplicate. And then last but not least is cytokinesis. So this is usually happening about the same time as telophase. So cytokinesis is just the division of the cytoplasm. So at this point, when we were in telophase, you now have two nuclei. Well, cytokinesis is going to break apart that cytoplasm so that now you have two separate cells. Otherwise, you'd have one cell with two nuclei. The two new cells that are created are going to be genetically identical. So that's really important. So mitosis creates two completely identical cells. So here's another view of that. So you've got interphase, normal cell, prophase. You're going to see those chromosomes start to condense. You've got your spindle fibers starting to form from the centrioles. Here you can see that nuclear membrane is broken up. Metaphase, you're lining up in the middle. Anaphase, you're being pulled away. Telophase and cytokinesis, you have your two new cells forming. This is one of those things that's usually easier to understand if you can see it flowing. So let's just take a look at this animation. So here's another version. You can see your chromosomes condensing. Nuclear envelope is disappearing. Your spindle fibers form. Line them up in the middle, then pull them away. And you have two nuclei forming, and then cytokinesis. So again, prophase, metaphase, anaphase, telophase, and cytokinesis. Here are some stained images of actual cells. So these are animal cells going through mitosis. Here you can see those chromosomes, the blue things, those are all condensing. Here you've got metaphase, anaphase, and then this would be kind of interphase, or telophase as it's reforming your two nuclei. Here you have some onion cells. These are like the roots of a plant cell. And so you can see here you've got early prophase where it's kind of grainy looking, and then late prophase where you can see all of those nuclei. I mean, not nuclei, chromosomes. Here's another slide showing mitosis. I believe this is a whitefish blastula. So there, that would be a really good example of metaphase. Here, you can see, let's see. Here you can kind of see them being pulled apart. So try to quiz yourself. See if you can identify the different phases in this diagram. So if we start at the top, this would be interphase, prophase, metaphase, anaphase, telophase. Again, try to quiz yourself. So this would most likely be anaphase because they're being pulled away from each other. This would be your centriole, and these would be spindle fibers. I'd say that one's going to be telophase because you can see the two nuclei starting to form. And this one would be a really good example of prophase. All right, see you in class.